Okay, good morning, almost good afternoon, everybody. Thank you very much, all of you, for joining this Pop Days 2015. Today is indeed a very special day for all of us because we are going to have the chance today to listen to 12 very brilliant young researchers from a very wide variety of fields and coming from 11 nationalities, so it's really a rich experience that we're going to share with them. It's also a lucky day for them, well, we hope so, because it's an opportunity for them to present their passion, their research, the work they've been doing yesterday and until very recently to improve their presentation, including the technical stress at the very end. So this is a great opportunity for you to share your science, to practice and to tell us if you will be able to have your grandmother or your nephew understand what you're talking about or what actually are you doing. So we're very excited to listen to you. And I would like to start with a very big applause because the exercise that you're going to do is not an easy one, and we would like to welcome you and by a big applause. So.
Bonjour. Thanks for having us all. For the most part, most of the time, my son Theodore's behaviour is really very good. But every now and again, he'll have a tantrum, and we'll have to deal with it. Volcanic systems are very much like toddlers because for most of the time, they're really lovely. But every now and then, they make a really big mess and can be very costly. Now, it doesn't matter anymore when an individual volcano erupts, because modern society is interconnected and it's growing. One of them will around the globe soon, at some point. So it will affect us all. What I'm doing is developing a new technique, which means that we can understand individual volcanoes far better than before. Like toddlers, they're all unique, they're all special, and if we are to respond efficiently, we need to understand them and also have the capacity to respond. But as volcanologists, we don't have the techniques that are fast enough to gather data as efficiently as we need to understand volcanoes on an individual level and then inform governments and society. My technique is 10 times cheaper and 10 times faster than conventional techniques. These are crystals that form and grow, they're born, they grow up, and they develop in different environments within a volcanic system. The key is in the chemistry. That's where the personality of the volcano is, and we can determine their mood swings. Because the technique is so rapid, it means we have a bonus here. Not only can we understand volcanoes in rich detail, we can, within one day, understand what's happening when a new eruption starts. And that's the big difference with this technique. It means that not only can we understand volcanoes' personality, but we can also understand and inform and protect modern society as soon as an eruption starts again. Thanks for your time.
How many of you have ever taken antibiotics? Modern medicine is dependent upon antibiotics, whether it's for a child who has an ear infection or an upset stomach or an elderly person with pneumonia. Antibiotics are not only important for treating infections, but also for people with reduced immune function, such as patients undergoing therapy for cancer, or HIV/AIDS, transplants, before and after surgery. Antibiotics are critical to the way that we do medicine. So imagine yourself in the year 2050. We're all a little bit older, and with that, our bodies are a little bit older too. What do you think would be the leading cause of death? Well, we expect that it will actually be due to antibiotic-resistant infections. In fact, we expect that in the year 2050, 10 million people will die as a result of these antibiotic-resistant infections. These are the bacteria that stop responding to our antibiotics. My name is Michelle Buckner, and I work on antimicrobial-resistant infections. I try and understand how these bacteria stop responding to antibiotics and what we can do about that. So what are antibiotics? Well, they were first discovered in the 1940s, and over the next 30 years, many new antibiotics were developed. But since then, very few antibiotics have been discovered. And what's frightening is that with that, we've seen an increase in the rise of resistance to every antibiotic we have. And this is where my work fits in. I've discovered a new technique and developed it that uses red and green fluorescent bacteria to watch the movement of these antibiotic-resistant genes. The type of antibiotic resistance that I work on can be thought of a little bit like Pac-Man. They're inside the bacteria, and they're able to chew up and degrade these antibiotics. And one of the neat things about bacteria is they actually form a sort of bridge between even distantly related bacteria. And across this bridge, genetic information, or DNA, is able to move. And that allows new bacteria to become resistant to antibiotics. And this is what I'm doing. I'm trying to stop this process from occurring. Now, the applications of this are very widespread. We could use this in hospitals, in agriculture, in sewage treatment, and even in farming. This work would reduce the amount of antibiotic resistance in the environment and reduce our chances of catching untreatable infections. And with that, I'd like to thank you for your time, and I'd be happy to take any questions.
Many of you will have had the problem that you turn on the light and nothing happens. Now, what could have gone wrong? There aren't all that many possibilities. So it's either the light bulb has burnt out or there is a power outage. And I can imagine that you will soon come up with an experiment how to separate between the two. Biologists, molecular biologists, have the same problem very often in their work. So imagine that some external stimulus comes to a cell receptor, which then passes on this information to turn on a response gene through an activator and inactivator. And you might have two different models for this system on the left-hand side and on the right panel. During my PhD project, we developed methods how to design optimal experiments so that you can tell the difference between these two possibilities with your experiment. So the way I see this is that there are many experimental biologists working on particular problems, and they try to dig deeper and deeper. Over the past decade or two, their work has been extended by the help of systems biologists, who essentially model biochemical systems in computers to help the experimentalists interpret their data. And where I come in is that I'm in a third layer trying to cut across all these fields by identifying commonalities and developing methods that help each one of them or the common problems that they have. It is a bit like business consultancy, where you try to disseminate the best practice across industries. So my background is mathematics. I studied mathematics, and my specialty areas are probability theory and differential equations. And now let me show you the two projects that I work on during this period that is funded by the AXA Research Fund. So one of them concerns model risk. Model risk is a notion that is probably known to some of you from financial mathematics. It is the risk of using the wrong model, even though you deliver the right results from your model. I try to do the same in systems biology. That is, try to find out if, due to missing data or unknown reactants or unknown reactions, your model is not an exact representative of reality, how much your predictions can be wrong. And the second project is trying to estimate contamination in so-called microfluidic devices that one of my colleagues develops. So these are small plates with thousands of little reaction volumes in it on a square centimeter area. He introduces 3 different types of DNA molecules into them and dyes them with the 3 colors, red, green, and blue. But sometimes he notices that there are clusters. Let's say there is a cluster of green colors here, and he wonders whether this is down to contamination, that is one reaction volume contaminating nearby ones. And I developed a mathematical model to be able to estimate this contamination rate. I combined percolation theory, which is a well-developed theory in probability, which means that there are these little white lines. These are connections. These are contamination routes. And I spread randomly red, green, and blue spots on the plate, and then see how, when you have a green spot somewhere across such a contamination route, then it contaminates its neighbors. And from the second image, I try to estimate how much contamination happens in the system based on the first picture. And I'm driven to help biologists help problems that otherwise they couldn't solve. They couldn't solve without mathematical modeling. Thank you.
Thank you very much, all of you. You allowed us to see the variety of the fields supported by the AXA Research Fund to better understand the risk related to our planets, to the human beings, and also to the animals, and also to support the researchers themselves. So this is quite interesting. But now I think it would be good if we had a few questions from the room. Are there any things that you would like to take the opportunity to ask to our grantees about their work?
It's a question for the volcanologist. Do you think we will be able to soon, or not too soon, being able to characterize precisely each types of volcanoes and then decipher when they will erupt and what damages they may cause, and then ensure people who are living around it against volcanic eruptions?
So that's the ultimate aim, absolutely, is to put back into society some better understanding of probability. But to do that, we need empirical fits. So geochemistry is nice because we can go back through billions of years of Earth history and to generate a detailed and rich understanding of the patterns of each of our little toddlers' personalities. And then when the next tantrum happens, we can say, oh, we can see this. We know what this is. And that's the basis for the prediction. And that way, we're in a far better, more powerful position to forecast. So we can't stop them happening. We can't put a lid on. It's only going to get worse if you do that. That happens in nature. It's not good. But we can respond a lot better than we can right now.
Maybe one question for Michel from my side. We were seeing frightening figures for the causes of death by 2050. For the human beings, can we estimate if it's mostly related to their own direct usage of antibiotics, like in medicines, or what is the part of indirect that comes through eating animals or things like that? I don't know.
So generally speaking, it's important to remember that when you take antibiotics, it's not you that becomes resistant. It's the bacteria that are inside your gut and inside on your skin and on all these different places. So normally when we see antibiotic-resistant infections in the clinic, it's coming from places like hospitals during surgery and places that are exposed to large amounts of antibiotics, which is what you see in a hospital setting, for example. So that's sort of the source of resistance, and we do think that environment does contribute. We know that there's a huge amount of antibiotics that are used in food production. For example, in the US, they use literally tons of antibiotic each year to make the cows grow bigger faster. And this is a perfect environment for resistance to develop. And then if you eat contaminated meat, there's a chance that there will be resistance on there. and bacteria are able to share their resistance. So even if resistance is developed in, say, a cow, in a non-pathogenic species, bacteria that doesn't cause disease, that resistance can then be transferred to a human pathogen that would cause disease. So, yeah, it is pretty frightening.
Thank you very much. Thank you. Are there any other questions from the room? Yes?
Hello. I'm really impressed by all your works. I have a question for you, Michel. I believe that to work on bacteria, you have to love them because they are really important for our environment, the ecology. They have a great, great role in everything. The resistance to antibiotics is really a huge problem for us, for everyone. I've known for sometimes that there's another solution that uses phages. So these are viruses that aims bacteria. And I believe that they have also a nice role to play. Do you have an opinion on these?
Yeah.
Thanks.
You're right, I do love bacteria. So what you're talking about is using viruses that are specific only to bacteria, and they can then infect the bacteria and kill the bacteria, which is a really great tool. And there's a lot of people looking at how that can be developed into something that we call phage therapy. One thing, though, to remember is that there will be resistance developed to these phages, because like anything, the bacteria and the viruses that prey on the bacteria are in an arms race. So they're constantly competing. So I do think that phage therapy is something to study. I think it's interesting. And I think that if we're to effectively fight the rise of antimicrobial resistance, we're going to need a lot of different tools in our arsenal. And I think phage therapy is one, and I think the approach that I'm using is another one.
Thank you very much, all, and thank you for your questions.
So the sun, as we usually think of, it's a big ball, yellow ball, iconic ball, very quiet, very romantic, as we could imagine during a sunrise. While actually the sun is way more active. Here you see the sun rotating and evolving in time with a lot of activities. This activity is generated from the magnetic field, which is popping out into the atmosphere, the solar atmosphere, that we also call the corona. We can see some very bright regions where the activity is very intense, but we can also see some darker regions that appear slightly more quiet, slightly more darker, while actually I'm studying those particular regions. Those regions, what we know is that they're large and that the magnetic field is open there, where the solar material can escape, a bit like a window, an open window where the solar wind can flow out of the corona. What we do not know is how do they exactly form, how do they evolve with time, and how do they vanish. This solar wind, this actually fast, constant solar wind, is blowing out of the corona and reaching the Earth. While reaching the Earth, it brings a lot of solar material, a lot of particles. Those particles can interact and hurt the satellites, or more particularly the solar panels, and can destroy and damage some of the solar panel cells. It can also disturb and interact with the technology and all the devices whereby our astronauts, it can reach the Earth and disturb the power plant, the power network. And another example is the communication. And for example, in plane company, in the future, we really want to learn more about this solar wind and the space weather that is generated. And for also the future, for the exploration of the future exploration of space. And I'll leave you with this image.
So Judith has just tell you about solar wind, but now I hope you have with you your warm jackets because the weather is going to get even more stormy. My name is Francesco Zuccarello, and I developed computers model to improve our capabilities of forecasting solar storms. Solar storms are huge, sudden explosion that occurs on the sun, and similarly to earthquake, generate tsunamis in space. While I'm sure you don't want to be walking on a beach when a tsunamis arrive, so do astronauts don't want to be out in space when a solar storm occur. But which is the origin of a solar storm? So this is a real image of a portion of our sun in which we can see a sneaky filamentary structure that, guess what? We call it a solar filament. When at times the filaments start to move and eventually result in big explosions. So congratulations, you are looking at your first solar eruption, real solar eruption. Now, what we want to understand is why this solar eruption occurs, and can we find some critical parameters that can be used as a precursor to study the solar eruption and therefore to forecast them. So what I actually do, I develop computers models that allow me to study the physics of solar eruption. So on the left, you might recognize our filament, and on the right, the purple structure is a model filament. Now, I focus, of course, in determining which are the critical parameters that will lead to an eruption, and what my model shows is that if the filament reaches a critical height, then the eruption will occur. So in the future, we will not only be talking about how is the weather in Paris or London today, but we can actually say how is the weather in the space today. And the most amazing thing is that we could answer windy, but with 50% chance of storms. And that's it. Thank you.
Okay, from the sun back to Paris, more ground to earth. I guess you were all super excited today to wake up in the morning and come to this special day. And I guess you dressed up, you took your bag, and then you took the stairs, and you found your way to AXA headquarters. 50 years from now, it will not be that easy for you even to take the bag and going down the stairs. It's going to be quite a challenge. And even worse, there's a good chance in 50 years you're not going to remember any of these experiences coming every day to AXA. You're not going even to remember you worked for AXA or even lived in Paris. And I know it sounds a bit sad, but this gradual loss of one's personality is what gives me the motivation to do my research. My name is Shahaf Peleg, and I'm working on aging and how to live healthier and longer. So I'm going to start with this very simple concept. You have here orchestra, which you can think about it as the DNA. And the DNA is composed from many genes, which you can associate with different musicians. And it's all very nice, but alone, they cannot do anything. They need a conductor to really orchestrate the orchestra to make this music in tune, right? And we know now, in aging, the conductor starts making mistakes, which is very bad for us. And how do we think it happens? So we know, for instance, in young animals, and I'm going to talk, I forgot to mention, I'm going to talk about flies as model, this little zzz thingy. And then we have in young energy production in the cells, and we have on the other end the gene orchestration. And the uniqueness of my project is try to bring these two subfields together. And what I found out that there is a messenger, or there are messages going from the energy production to the gene orchestration. And in young, don't worry, everything is fine. The problem is as we reach midlife, something around 40 or 45, the energy production starts to change. It actually starts to increase for unknown reason. And as a byproduct, the message or the messenger is becoming stronger, which is bad because it makes the genes go out of tune. It affects the conductor. And that in turn creates cellular damage and also memory impairment. Unfortunately, for my talk, I cannot focus on this. You can come to me later and talk about it. I'm going to talk about just physical damage. I'm going to illustrate it in these two movies. You see on the left, there are young flies, and here they are midlife flies. We knock them down, and we measure how much time and how high the flies can go up. And you can see the young flies really get the task very easy. They go up quite high. The midlife are really lazy. They stay really down. The main point, can we help the mid-fly? Are we going to give up or are we going to try to do something? And the idea that I had was to actually, it's very difficult to target the energy production or the orchestration, but we can target the messenger. So we can, genetically or with drugs, that's what we are testing, we can try to reduce the effect of the messenger. And now if we compare flies at the same age at midlife, these are normal flies, and these are flies where we reduce the messenger. You can see that the physical activity is increased. And if you bring it to the AXA world, you can say, these two flies pay the same money to AXA, they are the same age, but these ones are probably going to use the AXA insurance much more often. Hopefully, it is my hope now to move a bit forward and try to translate this knowledge that we acquired from the flies, design drug targets so we can translate it for us human, and hopefully in the future it can help us at least to solve some of the age-related problems. And I just would like to acknowledge everyone and also our two wonderful coaches. It was great 2 days. I did great, right? Okay. And thank you very much for your attention.
Thank you all. I hope the AXA employees don't feel like flies in a bottle. I think it was, again, very interesting. Are there any questions from the room that you would like to ask, either on the weather coming from the sun? Yes?
Hello, I'm Amandine, working for at AXA. I have a question regarding solar storms. I was wondering whether windstorms, the structure of the storm, would look like solar storm. Are there correlations or similar structures?
Well, not necessarily, because these storms are actually a huge, very fast flows of charged particles. So you have to imagine that they expand greatly from the sun to the earth. And in fact, if you noticed in the first image, you would see a little blue dot. That was the Earth. So it's a kind of different aspect. And it's also very important. The main difference is these are storms that also contain magnetic fields. So their dynamics is different. It's just more complex than the dynamics of storms on Earth.
And is it similar to any other structure, or is it a unique structure?
Well, let's say it's somehow, it's a fully developed complex three-dimensional structure that is not that easy to imagine. But imagine like a huge, huge cloud containing charged particles instead of water. And then this cloud is actually moving. So it's not the cloud putting the water, it's all the cloud moving towards the Earth and hitting the Earth. So it's like if you're in a plane and cross a thunderstorm, actually. This is, I guess you will feel something similar to that.
Thank you.
I have a question. Do you think your research could be interactive with the ones from Matthew? Because you're working on on eruptions, solar heat, and many similar things.
Well, in fact, there are definitely common points, for sure, from a mathematical modeling point of view. Because, for example, the example that I did with earthquakes is just not by chance. In fact, the mechanisms in which earthquake occurs, so the mathematical model, the catastrophic models that describes earthquakes, it's actually the same models with different equations, but the same principles that we use for our model of eruption. So, of course, I guess that we could have and develop some synergies at a certain level, let's say.
Okay. I have a quick one for Judith. What's the time between the moment that the wind happens on the sun and we may feel it on Earth? Can we say that?
Yeah, so the fast solar wind goes 800 kilometers per hour, between 700 and 800, so it's few hours. So when the coronal hole is on the side and then it's slowly coming, we see coming with the sun rotating, we know that in few hours it's coming toward the Earth. And we cannot shut down the window, but we can behave in different way.
Okay, quite short, huh? Indeed, yeah. Okay. Are there any other questions? Yeah?
It's a question for Shahaf. It was really interesting to understand how the messenger is working. From an epigenetic perspective, how can we, all, but even the flies, try to reduce the size of the messenger? Should we eat healthier? Should we send the flies to the gym so that the messenger gets low? Can we have an impact on the size of this messenger?
It's a very good question. It's a bit complex to answer, I think. It's definitely has been shown before that if you, for instance, use caloric restriction, where you somehow reduce your metabolism, it's beneficial for living better, healthier. In monkeys, for instance, you have loss of hair, which is good, I guess, for me. So now there are some studies that try to link it epigenetically. That, as you mentioned, the messenger is a byproduct of metabolism, which affects the DNA architecture. So I think, I guess it's a good tip for everyone to eat maybe a bit more healthy. It can actually reduce a lot of, or prevent a lot of the problems that we acquire in midlife and longer. It's a very good point.
So thank you very much all.
Well, hello everybody. I hope you're not too hungry. Hopefully the information that I'll give you will give you a better idea of the environmental situation that we're looking at. So maybe some of you know, maybe others of you don't. In soils and the microbial communities that live inside soils actually provide us with very many free services. And this is a very good thing given the current economic crisis. No? Some examples of these free services that we get from soil bacteria include water filtration, waste disposal, and plant fertilizer. Unfortunately, we have dumped quite a big number of substances in the environment that actually threaten the health of these communities. And my job as an environmental engineer is to try to protect them. Now, the substances that we put into the environment are already there. And we can't actually go back and remove them. But one thing that we can do is try to control their spread so that other communities that are not affected continue to be decimated. The substances that I'm talking about are particles. And you can kind of think of their behavior to be a little bit like a barbaric, When they're contained, you can mitigate the damage that they do. But if you release them, open the gates and let them go wild, they go from community to community pillaging. In soils, changing the pH and changing the ionic strength of the water, as well as changing the flow at which the water flows, is a little bit like opening the gates for these pillagers. So what we're trying to do is understand some of these environmental effects that essentially do this gate opening for the microbial contaminants. And we're trying to do this in order to, first of all, identify the concrete events that cause this, but more importantly, to quantify how open these gates are in case any of these events actually occur. The way that we're doing this is by looking at processes that happen inside a soil. And as you might imagine, it's very, very difficult to actually look inside a soil because it's opaque. So the neat thing that we've been doing is we created a transparent soil that actually allows us to literally see right through it. It allows us to optically access all the pores within the system. And with a set of cameras, we can actually spy on these particles. And we can measure where, how quickly, and when these particles move through the soil, when they get stuck, and when they get released, every time that we apply one of the changes that I just mentioned that open the gates. Now, the main outcome for the work that we're doing is to be able to pinpoint the worst set of conditions that open the gates for these contaminants to move around and affect different communities in the soil. But the ultimate goal is to be able to use this information to inform land management strategists about the conditions that they should regulate in order to actually try to keep these gates as tightly shut as possible. I'd like to thank you for your attention and take any questions.
We like good weather, hot and sunny. Mosquitoes also like good weather. However, we don't like to share our life with mosquitoes for two reasons. The first one is that mosquito bites us, which is very unpleasant. But the most important reason is that mosquitoes transmit pathogens. For example, dengue virus, chikungunya virus, which kills many people every year in the world. Actually, there is no effective vaccine to protect people from these pathogens. So one of the ways to protect people is to control mosquito populations. To control mosquito population, we use insecticide treatments, and they work in some cases in the control of vector-borne diseases. However, extensive use of insecticide treatment has led to the selection of resistance. So resistant mosquitoes are now present in the field population, and it is very difficult to kill them. What we know is that resistance affects the biology of mosquitoes. For example, resistant mosquitoes have a very reduced longevity and lower fertility compared to non-resistant mosquitoes. But what we don't know is the effect of insecticide-resistant genes in the transmission of viruses. So I am Celestine Atyame, and the aim of my project is to know whether insecticide resistance affect the transmission of viruses by mosquito vectors. The result obtained in my research will allow developing new tools to control mosquito population. They will also help refining the evaluation of risk of emergence of and spread of virus-borne pathogens in human populations. I would like to thank you for your attention.
A few words come to mind when we think about cancer. Cancer is a very ageless disease. It affects both old and young. It's also a very deadly disease. And even more, it's a debilitating disease. Luckily, we have chemotherapy, which we treat our cancer patients with. But then, unluckily, it's still incurable. Now, why is it still incurable? Because almost all cancer patients relapse. Now, why do those patients relapse? Because the cancer cells become drug resistant to all known chemotherapy. My name is Eline, and my aim of my work is to block this drug resistance. Now, I work with a plasma cell model, plasma cell cancer model. The plasma cells, they thrive in the bone marrow, where they interact with all the surrounding tissue. Now, how can they interact with that? They either secrete proteins, termed cytokines, to then receive from their environment, again, growth factors to grow and be protected, or they can secrete vesicles. Now, these vesicles split off of the cell membrane, carrying proteins that are inside the cell. You can look at it like small oil droplets that come off of a big oil drop. Now, imagine the bone marrow environment as a village with all healthy, happy people running around, and then suddenly this big, angry giant comes along, and he's very hungry. And so what does he do? He first starts sending threatening letters to those villagers demanding, "Give me some food." But if that's not sufficient, he also starts sending out postal packages. And those postal packages make those villagers give the food. And not only that, but also protect this giant against its hostile environment. Now, we don't know exactly what's inside that postal package, what makes those villagers actually give the food and protection. We do know that these vesicles, that they contain DNA, RNA, lipids, proteins. And in my project, I wish to identify exactly which protein or exactly which lipid is making those villagers give the food. And apart from that, I'm also looking at the postal service, the secretion machinery that's responsible for secreting these vesicles. And if we can block this postal service, we can then block the communication between the cancer cell and its environment, that way making the cancer cells more sensitive to the chemotherapy and ultimately lead to an improved outcome of the cancer patients. Thank you.
Thank you very much. We're seeing really very interesting imagery and very nice ways to make very complex things understandable, and we thank you all for that. Are there any questions for the room for three researchers?
I have a question for Celestine. Do the environmental conditions have some effect on the transmission of the virus or the disease from the mosquitoes? For example, in Africa or in Martinique?
Yes, there are some studies that have developed in the lab showing that temperature affects the transmission of virus by mosquito. If you increase the temperature, you increase the transmissibility of the viruses. This is the most important factor that is analyzed in the laboratory. It can also have the effect of humidity. When we compare the transmission of pathogen between countries, you have a lot of other factors that can be involved. For example, you have a factor involved in mosquito genetics, in virus diversity also. So the transmission of pathogen depends on complex interaction between virus, mosquito, environment, and also host, like human and animals. I don't know if I answered
Yes,
your question.
it was a really interesting question because I'm from Martinique and some people from my family was bitten by mosquitoes, and we were really thinking about how we could treat that question, because I have some friends who went to Africa too, and they were bitten by mosquitoes too. Then we were.
Yes, sometimes you don't have the same mosquito. For example, one species of mosquito do not transmit every viruses. So, for example, in Africa, you have mosquitoes that transmit malaria, and in Europe, these mosquitoes are not present. So you have the genetic diversity in mosquito population. We also have different viruses in different countries. So our work is to understand which kind of interaction exists between these different partners, and in order to evaluate the risk of the emergence of epidemics. So for example, in Martinique, when you compare mosquito population in Martinique and in France, you don't have the same mosquito populations. And we also have adaptation of virus according to environment. For example, chikungunya virus that generate epidemics in La Réunion in 2007, and chikungunya virus that generate epidemic in Italy in 2010. You have a mutation between these two viruses, but it's not the same virus that generates two different epidemics.
So it is very difficult to define a good treatment for this.
Actually, we don't have vaccines, so the most important work is to focus on mosquito by developing new tools, by educate population and tell population that you must clean the water container very close to your habitat, because mosquito lives in the water in the first stages. We have larvae living in the water. So you must be careful about the environment around your home, around your city, to be able to reduce as possible the density of mosquito before hoping to get a vaccine or very efficient treatment.
Thank you.
I have a question regarding the cancer research. My thought after hearing all of you is that these mosquitoes, cancer and bacteria get resistant to all the things we use, but we do not get more resistant. And it's quite the same question. Is there any link between what we eat that could make us resistant? And I know that for cancer, there are a lot of studies that there is this link.
Well, basically, it's true what you say. Whatever you eat and things, or what you do with your body will have an influence on all the cells in your body. Of course, to make them susceptible to cancer, there's almost always a genetic origin in the way that a healthy cell changes into a cancerous cell. And if you eat things or do things that is harmful for your cells, then they will react in a way that could make them cancerous. So if you are in a lot of sunlight, the sunlight will affect the DNA in your cell, and in response, the DNA will change and then could become a cancerous cell, which is similar to eating things. To think if you can eat something which makes your body more resistant, I guess you could. I guess since we do try to make therapy, in my case, to try to find something which blocks the secretion of these vesicles, if you could find something in food which would also have that same effect, blocking this secretion, I guess then in theory that would be possible. It would be then that you're actually not needing drugs, but something natural which mimics the effect of the drugs.
Thank you. Can I take just a quick one to Veronica? Can you tell us a little bit about your transparent soil? What is it made of? How do you make it work?
So the transparent soil is actually made of a material called naphion. It's a material that is typically used or was designed to make fuel cells. So it's a permeable electron membrane. The nice thing about naphion is that it has a refractive index that is very, very close to that of water. So when you submerge it in water, it actually matches the refractive index of the liquid, and that's what allows us to see exactly right through it. So when we shine a light through the transparent soil, it doesn't get distorted, and we can actually just follow the particles that we're interested in and see where they're moving in the space.
That's brilliant. Thanks. Thank you very much. Okay, so thank you all.
Good morning. This is the world's fastest train. I'm sorry to tell you it's not the TGV train, but it appeared in all the newspapers some months ago. This is a Japanese magnetic levitation train that broke the world speed record with 603 kilometers an hour. And just in last April. And this was possible thanks to materials called superconductors. So my name is Isabel Guillamon. I'm a researcher at the Universidad Autónoma de Madrid in Spain, and my job is to understand how this superconductor works and how to make them much more efficient. So this fastest train is just one of many applications of superconductors. Superconductors are, for instance, the reason why you can take medical images without damaging radiation in hospitals using magnetic resonance imaging machines. So let me tell you what a superconductor is and how it differentiates from a normal conductor. So a normal conductor is nothing else than a material that can carry electricity, and it happens because electrons can move through the conductor. Imagine that electrons are people, and their motion creates a current. So in a normal conductor, electrons behave as people in a very busy day in the corridor, in the metro corridor. So they move in a very disordered way, so they pump each other, and they reduce the current carrying capabilities, consuming power. Instead, in a superconductor, electrons move like people working in a very synchronized way. So it's like a perfect dancing. So as a result, electrons lose the electrical resistance and can carry electricity without any losses. So today, 20% of the electrical energy is lost between the power plant and the households because the electrical resistance of the cables. So think of having superconducting cables with zero resistance. So we could build much more efficient motors, wind generators, or computers. We could transport and storage zero emission energies and make them much more cheaper. So if these superconductors are that great, why don't we use them more? So the main reason is that a small tornado called superconducting vortices appear within superconductors. When under the action of a current, these tornadoes move, and they destroy people. They destroy this synchronized electron motion, and as a result, they produce non-zero resistance and kills the superconductivity. So for that reason, it's very important to stop this vortex motion for any application of superconductors. So just to give you an idea of the problem, a superconductor with a size of 1 cubic centimeter, that's small, we can find 50,000 millions of vortices of this small tornado. So my research aims at finding the best ways to stop the vortex motion. So for that, I used very powerful microscope that allow me to visualize vortices one by one. and nanofabrication tools to manipulate matter at the nanoscale and create effects that can effectively pin down these vortices. So hopefully we will find new methods to stop vortex motion and to improve the current carrying capabilities of the superconductors and achieve the dream to create a new amazing electrical world. Thank you.
I am sure that all of you have spent a lot of hours stuck in traffic. And I know that time spent in traffic is not only time wasted, but time miserably wasted. So our research is on transportation systems. And of course, I cannot promise you that we will going to solve the transportation problem, but we hope to obtain a better understanding of a transportation system, and this will be the key to design optimally future transportation systems. What we want to do is to answer the question, which mean of transport a commuter should choose if he had several choices. What is new in our work is that we model our transportation system, we analyze our transportation system in a micro level, taking into account all the fine details. Of course, there are several things that make this analysis very demanding and not very easy. The first thing we should take into account is that the decision of a passenger is affected by the decision of all other passengers. Imagine that you have to choose between metro and car. If you know that all other people will use their car, then you conclude that there will be a traffic jam and you had better choose the metro. So every day, in every big city, there is a game that takes place among all passengers, where every commuter plays against all other commuters, and we can handle this situation using game theory. The second thing we should take into account is the uncertainty. In a transportation system, travel times are random, waiting times are random, and also the free space in a transportation facility is also random. So you want to go to the AXA headquarters, you use line 9, but you are never sure that the next metro vehicle will have enough space for you. We can handle all these uncertainties using queuing theory. And the third thing we should definitely take into account is the effect of information on customers, on passengers' behavior, as nowadays more and more applications are popping out that provide commuters with fairly accurate delay information. So hopefully, this micro-level analysis will help us to predict future travel demand and design optimally future transportation systems. Thank you very much.
During the last century, global fish stocks have been exploited and depleted to such low levels that, sadly, we might be the last generation to be able to obtain our food from the ocean. So why are the fish stocks collapsing? Why aren't they recovering, even if we stop fishing? What is causing this, and what can we do to avoid this? In my research, I show that a relatively controversial phenomenon called fisheries-induced evolution might explain, or at very least might help us to understand why our fish stocks are collapsing. So here we have a nice, diverse fish population, which consists of different-sized fish. Now, when we go fishing, we typically target the largest individuals. We like to cook a large fish. We like to eat a large fish. If we have to sell a fish, we get a better price for it if it's large, so it has a high economic value. And most of you who have been angling, for example, they do remember how it feels to catch a big fish. It's an experience you don't easily forget. So when we are removing the large individuals from the population, we are not just removing the big fish, but we are actually removing genetic material from the population. So the genes that determine the large body size, so the large size genes, are removed. The small size genes remain in the lake or in the ocean. And these large size genes are now permanently removed. They might not actually come back. And this is what is fisheries-induced evolution. In my research, I have shown that if we catch the largest fish out generation after generation from the population, we do induce genetic changes in these fish populations. So we do actually change the sequence of the DNA, which you can imagine is not very temporary change. It actually might take quite a long time to reverse. It might be even irreversible. But I also show that only after 5 generations of fishing and catching the largest fish out, we actually induce gene expression changes in over 4,000 genes. So why is this important if we remove or reduce the genetic variation in a population? Well, imagine a French vineyard, which would consist mostly of grapes that are very similar to each other. So they are genetically and phenotypically very similar to each other. Now, imagine there is 1 year, there is a very harsh winter and a lot of snow, or there is a pathogen, some mildew that affects the grapes, and it kills all the grapes in France. Pretty bad, huh? There won't be any wine made in France that year. So this can be compared to similar situation in the fish stocks. If our fish stock consists of very similar, genetically and phenotypically, very similar, identical individuals, this reduces their resilience towards environmental disturbations. So harsh environmental conditions or some pathogen might actually wipe out what's left of the population. And therefore, we have to care for this genetic and phenotypic variation. Now, what can we do then about it? We can't stop fishing. I mean, fish are a lot of people's food source, and a lot of people's livelihood depend on fishing. We can't just ban fishing. But what we can do, we can fish more sustainably. So first of all, we should fish less. And second of all, we should fish less selectively so that we can protect and conserve the genetic and phenotypic variation of our fish stocks. Thank you.
Thank you very much. So lots of thoughts and inspirations for our future behavior, be them the transport we're going to take or the fish we're going to eat or eventually not eat at all. So are there any questions from the room to our researchers?
Yes. Good morning, and thanks a lot for this very interesting presentations. I have a very naive question regarding the fishing around the world. You were saying that there is a way to promote more selective fishing, and to my knowledge, at least the way I imagined fishing was that actually the problem is that we simply take out everything that's around on the that's simply around. Like very big boats throw their nets and they take out sharks, dolphins, fish, you name it. So how can we make this fishing more selective, as you're suggesting?
Or actually less selective. So we should avoid being very selective. So,
exactly. Sorry, I wanted to say that.
Yeah, exactly. So you're right. That's why I mentioned the first point: fish less. That's where we have to start. We have to fish less and not just vacuum all the oceans from the fish. It's the same thing in a smaller scale. For example, in Finland, where I come from, we are trying to prevent that we could release the largest individuals back to the lakes when we catch them, to see that, okay, this is, we call them BFFs, so big, fat, fertile females. So we like to release them back to the lake. If you catch one, please just put it back. But as long as we are gill netting, we are using gill nets to fish, that doesn't help anything. You can't just release a fish that's been tangled in a gill net for a day or something. So therefore we have to think about our fishing effort and cut down that first. There is a way, of course, then when we talk about commercial fisheries, we have the problem of side catch and all that thing, and this, that we vacuum basically all the species that we have. There are methods and there are ways, I can't go into specific, but I know there are ways how they could reduce the selectivity and actually try to target, for example, the middle-sized fish, not the small ones, because they haven't reproduced yet. If we take them out, then they are not going to help the population productivity in any way. Not the large ones, because they are the valuable ones. They are the ones that actually reproduce the best, among other things, and of course then we conserve the variation. So it has been suggested that either we fish non-selectively, so we just pick whatever randomly out of the population, or then we try to target both the large and the small, or just the intermediate-sized fish. So I would say that a wise man once said, there are 3 ways to do it. Fish less, let everybody spawn at least once, so don't take the very small ones, and then protect the large individuals.
Thank you. That's quite an unusual saying, to protect the big ones, but we take it. Athanasia, I was wondering if in your research somehow you would consider the trust of the customers towards a given transportation system. And for example, in the context of driverless cars and maybe, who knows, driverless buses or whatever, is that something that's being taken into consideration?
Yes, it is taken under consideration. We have some parameters that reflects this trust to service providers. And there we also have some types of consumers, sometimes of commuters. They can be less or more trusty, and they can trust more or less the service providers.
One more question?
Yes, just I have one question for you, Isabelle. Regarding superconductors, which seem to be extremely promising in the type of applications that they can bring to us, are the vortices you're working on kind of the last blockers before we know how to use this in an efficient manner in let's say, and apply it? Or are there other type of constraints that maybe other researchers are working on? And following that, my question would be, how quick do you estimate that we will have solved or learned how to deal with them in a fairly, realistic manner?
Okay, so that's true. There is another important constraint that I didn't mention, is that superconductivity is a property that appears in some materials when they are cooled down to low temperatures. So with low temperatures, I mean minus 150 degrees. So materials need to be cooled down to become superconductors. But that is not that expensive, so that sometimes is, the gain that you obtain from that is a lot higher than the cost. So there are people trying to work in parallel. There are 2 open problems. One is to try to find materials with become superconductor and higher temperatures, and then to make they work more efficient, just to trying to pin down vortices. So there have been recent discoveries of new materials with much higher critical temperatures. So the investment in superconductivity is increasing by a lot, and the application of superconductivity they are increasing by a lot. So I think that they will continue making a new improvement and new development. So expected that new application appears in the next few years.
Thank you.
Welcome.
So thank you again for our last 3 sets of researchers. And it's time for the big thanks for everybody. So for you, the audience, and for the questions that you've shared with those researchers. For the 2 coaches who have done tremendous job in training those researchers, I think they deserve a big applause. Isabelle, I don't know where she's hiding, who's been the master of the Pop Days together with the communication team. And so thank you, everybody. Please join us, all the researchers, for a big group photo. And please take the time of the lunch to enjoy, discuss, exchange with our researchers. Take their booklets if you haven't taken them yet, so you can have the contacts, you can follow them on Twitter, you can get more information on their websites, and of course always stay tuned with the AXA Research Fund, and thank you very much to all.